Hello everyone, welcome to my channel. I am Mariana Mihailidou, and in this podcast I will be getting in your genes. So be prepared because this journey we're embarking on is destined to be genome defining. Now let's not keep you waiting and dive straight into today's topic, which will be about the genes, right? Well, not exactly, because today we're exploring something even more intriguing. Junk DNA, or non-coding DNA, as modern researchers have so kindly decided to call it. And you might be wondering, what's junk DNA? At this point, I think it is best we begin with another question. What is coding DNA? Coding DNA is a part of our genome that encodes proteins that can therefore be transcribed and translated in order to produce the molecules that define an organism's structure and function. Long story short, the invaluable part that defines every single biochemical or structural characteristic we possess. In contrast, junk DNA is the part of the genome which does not code for a protein. It was once thought to have little to no function. This assumption, however, turned out to be a rather significant oversight. Recent discoveries have shown that these regions can have important roles in gene regulation and genome stability. This mystery that has been surrounding junk DNA started to unravel in 2012 when the ENCODE project, a major research initiative aimed at mapping all functional elements in the human genome, concluded that around 3/4 of the non-coding DNA in our genome doesn't undergo transcription, meaning it is involved in the creation of RNA molecules which are crucial for gene regulation, even though they do not directly contribute to protein synthesis. Furthermore, comparative genomic studies, which involve comparing the genomes of different species, have shown that some of this junk DNA is highly conserved, sometimes over hundreds of millions of years, which suggests that these non-coding regions have undergone positive selection throughout evolution and confer an advantage in terms of preserving or minimizing certain biological traits. Seems like junk DNA is not useless after all, right? So this brings us to the big question. What does junk DNA actually do? First of all, there are non-coding DNA sequences that are crucial to the expression of genetic information, such as the ones that carry the information for the creation of RNA molecules other than mRNA. These include the transfer RNAs, which, yes, transfer amino acids to the sites of protein synthesis, or ribosomal RNAs, which partly compose the ribosomes. Other RNA molecules, mostly unknown by the broader public, contribute to the regulation of gene expression. For instance, microRNAs bind to messenger RNAs, the world-famous mRNAs, and prevent their translation or promote their degradation. Studies have shown that some other sequences of the so-called junk DNA also play a significant role in regulating genetic expression by interacting with promoters. More specifically, silencers repress the transcription of specific genes, while enhancers, guess what? Enhance it, even though they are often located afar from the genes they regulate. They basically have the same function as dimmer switches. Instead of regulating light, however, they regulate gene expression. Interesting, right? So, you've seen how non-coding DNA influences gene expression, but its functions don't stop here. Another surprising function of non-coding DNA is in fact stability. Have you ever wondered what keeps our chromosomes from unraveling during replication? Telomeres, sequences of repeating nucleotides, form specialized DNA segments at the ends of all chromosomes. These are important in preserving the chromosome's structural integrity during the process of DNA replication by keeping the ends from being degraded. Without them, our chromosomes will start degenerating exactly like shoelaces that have lost their protective cover. And that, as you can imagine, couldn't be beneficial. Repetitive DNA, also called the satellite DNA, is also found in centromeres, the vital structures that link the sister chromatids during cell division. It is also present in heterochromatin, a disrepacted form of DNA, which plays a protective role by preventing harmful recombination and ensuring the correct separation of chromosomes during cell division. And because nothing in biology makes sense except in the light of evolution, as stated by Theodosius Nobzhansky, I cannot overlook the contribution of non-coding DNA in creating genetic diversity. You see, there are mobile DNA sequences, the transposable elements, which can relocate in a genome, inducing mutations or even regulating gene expression. Hence, they promote recombination and create new genetic combinations, leading to genome evolution. And here we are now at the most significant part of today's podcast. Can these discoveries be utilized for a cause other than satisfying our curiosity? The answer is yes. Scientists have linked these non-coding sequences with the risk of various diseases, such as breast and prostate cancer, autoimmune diseases, diabetes type 2, Alzheimer's, schizophrenia, even obesity. When discussing the association between cancer and non-coding DNA, researcher John Quankenbush explains that a gene alone cannot determine whether someone will develop cancer or not, In fact, there are millions of other variants lying in the so-called junk DNA, which can target genes expression up and down, and while each of these variants alone has a tiny effect, altogether can impact us significantly. The power of team effort, right? According to the scientists, this discovery could further be used for a more precise calculation of cancer risk and the determination of the most suitable type of therapy. Deciphering non-coding DNA would pave the way for groundbreaking treatments tackling the aforementioned diseases. Imagine if we could use the already existing gene-regulating technologies in order to identify a sequence that increases the risk of breast cancer by probably regulating a gene and correct it, restoring normal gene function. Or if we could apply RNA-based therapies to silence harmful non-coding RNAs involved in Alzheimer's and epigenetic therapy to correct abnormal gene expression in diseases such as schizophrenia or multiple sclerosis. The possibilities are endless. However, we have just started unraveling the secrets of the so-called junk DNA, and even though significant progress has been made, much remains to be accomplished. Our podcast now must come to an end. I hope that I restored the long-lost reputation of the misjudged junk DNA and that I awakened the investigative spirit that is hidden in each and every one of you. And please don't be shy. Any corrections and recommendations are very well welcomed. Now, while goodbyes can be tough, stay tuned, because we will soon recombine our alleles and delve into more fascinating genetic topics. See you in the next episode!